Once again, congratulations for successfully completing the workshop.
Thank you very much, sir. It's all possible because of support from seniors and then colleagues in the institute and blessings of elders like you, sir. Thank you very much. It was a big challenge, but it was a learning experience for all of us because for the first time we conducted it in hybrid mode, not in online mode, not in live mode, but in hybrid mode. And I'm happy to say that more than 300 people participated offline and another 200 to 250 participated online. I think that should be the trend in future also. Invite a limited number of people for offline and have maximum participation of people from online. And entire technology required was sourced in-house only. Dr. Sailaja managed it really well.
Very nice. Very nice.
And their hall committee, they managed it really well, sir.
Coincidentally, I was in Dehradun that time and I could briefly connect it for about 1 hour just before the session on that varietal identification. I mean, the non-varietal identification program. Just before that, I could connect for about 1 hour and then I lost the connectivity there.
So we are almost final with that performance, sir. Very shortly, we had invited inputs from all concerned, including
very good
experts in crop production and then protection. And already some 5-6 technologies which have been tested for more than 3 years in ICAR-IIRR, they have come for identification. And a set of technologies have also come from a cooperating scientist for testing for the next 3 years. So hopefully, just like varietal release, CVRC and then Gazette notification or SVRC notification, this will also come just like our NBPGR certificate, which will be signed by the coordinator or director along with DDG. We'll issue a certificate highlighting that so-and-so developed these technologies. This particular technology is suitable for this particular ecosystem. That's a
very good development, actually. That's a very good. I hope the private sector is also part of it.
They are now aware about it. I've requested them to nominate a few technologies, and then we can collect some testing fees also out of it.
Right.
That's the idea, sir. Mahendra sir, maybe another 5 minutes we'll wait and then start.
Prasad garu, good morning. MS Prasad.
Sir, good morning.
How are you? Fine. After a long time.
Seeing after a long time.
Yeah, after a long time.
But then special trip to Dehradun or any other assignment, sir?
No, personal trip only. We were there away for not only Dehradun, we went to Mussoorie, then near Gangotri, then Badrinath. We all about 15 days tour. I could see the terraced fields ready for sowing there. Rice. So I think maybe after 1 or 2 months, it will be very good to visit those places.
This time our director permitted us to go for even during sowing time as well as in the flowering time to visit so that we'll get a good chance of visiting many cooperating centers so that we can also monitor properly.
Yes, I think that is that's another development much needed actually. We should visit at least 2 or 3 times during the crop season If not all the ecosystems, at least few ecosystems, if we can monitor that way, I think we'll get better information. And also, I have forwarded one news item on the seamless farmers database. I think that's a very good.
That's a very good suggestion, sir. And I will highlight that when I discuss with our extension people and also with Dr. Sailaja. And I'm also waiting for Dr. Sheikh.
And also,
yes, sir.
Yeah. And also, Dr. Sundaram, I think government is going to come up in a big way to support that. That is the reason if we can take the initiative and show them some data already, then I think you can even have a long-term project, particularly with ECRIP around. You can make it a part of it from the research point of view. See, the government will collect from its own viewpoint, but from the ECRIP point of view, definitely it is a very good move.
Sir, I had a discussion with Dr. Laha. I understand that for the last almost 25 or 30 years, we already have a database of farmers from whom production-oriented survey data is collected. And then we have something in the form of a primitive database already. What we had to do is, maybe this year, when production-oriented survey is collected, we need to collect the GPS coordinates also, along with, now even a simple smartphone will do it. The data has to be geotagged for each and every farmer. That's the way we are looking at it. Together, we will see that how best we can go ahead with the targeted objectives, sir.
Yeah, the only thing is you need to take more steps towards authenticity. See, somewhere you have to involve either a state department or a central department organization, so that that can be made more authentic, and it can also support the government, and ICAR can again take because it will come to the credit of ICAR that it is helping the government in a big way.
Sir, recently you must have seen we published it's like a policy advocacy book based on production-oriented survey. We have made some predictions and also presented the changing pest scenario particularly. And I'll send a copy of it to you, sir. It was recently released in the ECRIP workshops. And for your information, ECRIP's name has been mutated. It has been changed from All India Coordinated Rice Improvement Project, which mainly covers rice improvement. It has now been changed as All India Coordinated Research Project on Rice, which will cover all the disciplines.
Okay. Research project on rice.
Sir, another 1 minute we'll wait, Mahendra sir, and then we'll start. It's about 10:04.
Padma Kumar, are you there? Padma.
Yes, sir. Very good morning, sir. And yes, sir. A very good morning to you all.
Okay.
Good morning, Padma Kumari.
Good morning, sir. Kati, sir.
How are you all?
She's our seminar in charge. She takes
I'm saying it. Yeah. Nice to see that SAR is, having regular activities. That is most important. Very good.
That is because of IIRR. We are
No, definitely. IIRR is a part of. See, IIRR is a part of SAR. There is no question about it. Integral part. Yes.
Sir, let us start, sir. People will.
Yeah. We can go ahead. Let's start it.
Padma, can I start, Padma?
Director, sir. Okay. Sir, on behalf of everyone, I welcome Dr. Vara Prasad Garu for today's session. I request our Director, Dr. R.M. Sundaram, to give his introductory remarks.
Dear Dr. Vara Prasad, Dr. Mahendra Kumar, Dr. Katti, heads of sections at IIRR, Dr. Praveen Kumar, Director, Central Coastal Agricultural Research Institute, Goa, and all my colleagues from not only from Indian Institute of Rice Research and also the colleagues from Society for Advancement of Rice Research. I extend a warm welcome to each one of you. As you all are aware, agriculture and climate change are internally correlated with each other in various aspects, as climate change is the main cause for biotic and abiotic stresses, which do have adverse effect on the agriculture of a region. Agriculture is being affected by climate change in different ways. For example, we are already seeing variations in annual rainfall and average temperature, prolonged heat waves, modifications in weeds, pests and microbes, global change of atmospheric CO2, and increasing frequency of cyclonic weather and fall in ozone levels, and also fluctuations in sea level. The threat of rapidly changing climate has greatly driven the attention of scientists, as these variations are imparting a negative impact on global crop production, thus compromising food and nutritional security worldwide. According to some reports, agriculture is considered the most endangered activity which is adversely affected by climate change. Rice yields are expected to fall by about 5 to 10% for every 1 degree centigrade increase in global temperatures. Rice is considered both a victim and also a culprit of climate change. This is because wetland rice paddies generate a significant amount of methane, but the crop also serves as a significant sink for carbon dioxide. In today's world, in the realm of a rapidly changing climate, food security and ecosystem resilience are the most concerning subjects. Agriculture needs to sustainably contribute to food and nutritional security and also rural economic development while reducing the negative impacts and footprints on the environment. Demand for sustainable agriculture, which is less dependent on external inputs and better suited to marginal environments, has revived interest in many traditional systems of cultivation with suitable contemporary modifications in order to sustainably increase the production of nutritious food and encourage dietary diversity. Climate smart agriculture is the only way to lower the negative impact of climate variations on crop adaptations and also increase resource use efficiencies and adopt strategies to mitigate climate risk in a system-based manner. On such an important and contemporary topic, today we have with us Dr. P.V. Vara Prasad, Professor and Director, Crop Ecophysiology, Sustainable Intensification Innovation Lab, Kansas State University, USA, to share his knowledge on the topic, crop responses to key climate change factors and need for systems research with reference to major cereals. I have listened to an enlightening talk by Dr. Vara Prasad during the recently conducted International Agronomy Congress. And I'm sure that today's talk will also be very motivating and illuminating us on a contemporary topic of interest to all of us. I welcome Dr. Vara Prasad and each one of you to the webinar and request my colleague, Dr. Mahinder Kumar, Principal Scientist and Head Agronomy, and also General Secretary, Society for Advancement of Rice Research, to introduce Dr. Vara Prasad. Dr. Mahinder, you can go ahead.
So thank you, sir. Thank you, Dr. R.M. Sundaram. Thank you, Padma Kumari Madam. And once again, I welcome all the delegates who are offline and online for this very important and very useful webinar. And this is the, I think, webinar series, sir. We are conducting Society for Advancement of Rice Research along with Indian Institute of Rice Research. On the occasion of Azadi ka Amrut Mahotsava, we are conducting this series of webinars for the benefit of the rice scientists across the country. We have more than 300 to 400 rice researchers we are under a CRIP program. They are all, I think, part of this society. They have been all invited. I am have very proud privilege to introduce our today's very eminent speaker, Dr. P. Vara Prasad Garu. He is a university distinguished professor and endowed professor in agriculture and director of Sustainable Intensification Innovation Lab at Kansas State University. He obtained his BSc and MSc from Andhra Pradesh Agriculture University, India, and PhD from University of Reading, UK. His research focuses on understanding responses of crops to changing environment, developing best management strategies to improve yields, providing food and nutrition security to smallholder farmers, and improving livelihood of people. He has active programs in multiple countries in Asia and Africa. He has published over 340 peer-reviewed journal articles and book chapters, trained 160 students and research scholars from 25 countries, and gave more than 100 invited talks in 40 different countries. He received more than $150 million in grant funding and donations. His research has been cited more than 18,900 times with an H index of 73 and I-10 index of 207. He is an elected fellow of American Society of Agronomy, Crop Science Society of America, and American Association for Advancement of Science. He serves on the International Commission on Sustainable Agriculture Intensification. He is the past president of the Crop Science Society of America. It is a great achievement. Sir, we are very thankful to you for readily accepting our invitation to deliver this webinar. I had Dr. P.V. Vara Prasad during International Agronomy Congress, as well as the other webinars which has been given by him. It is really, for everybody, it will be very enlightenment. We are all welcome you, sir. Now the floor is open for you, for your talks. Thank you, sir. Thank you very much. Thank you, one and all.
All right, good morning, everyone. Thank you so much. Hopefully you're able to hear me and also see my screen in full view.
Yeah.
Is it on full view?
Yes, sir. Yes, sir.
Okay, excellent. Greetings again. Thank you so much. Thank you, Dr. Mahendra Kumar Garu, for giving a nice introduction, Principal Scientist and Head of Agronomy, and also giving me this opportunity to speak today. Thanks are also due to Dr. Padma Kumari Garu, the Principal Scientist and Chair of the Seminar Organizing Committee. First, I would like to also thank Dr. Aram Sundaramji, the Director of the Indian Institute of Rice Research. I met him last year during the International Agronomy Congress, and I'm really excited and glad to be part of this seminar on the occasion of the 75th anniversary of our Indian independence, Azadi Ka Amrut Mahotsava. It is such a great way to celebrate through national and international seminars, which are useful to the scholars of not only India, but also around the world. I had the honor of doing a few of these at different institutes. On this occasion, I would like to congratulate first the food producers, the farmers of India, for doing such a great job of producing enough food grains to meet the growing demand of increasing population of India. Congratulations to them. This was also only possible due to the hard work of scientists and the great innovations that were developed by many institutes of ICAR, Indian Council of Agricultural Research, and the state agricultural universities. And I'm sure that Indian Institute of Rice Research has a lot to contribute to that as well. Considering the short time today, in the next 45 minutes, I will be focusing on climate change, which is very important, as Director Sundaramji told. But I'll only cover the two aspects of climate change, the increasing temperatures and the carbon dioxide, to show the impacts on grain crops. There are other climate factors which are becoming more critical. That includes drought, UVB radiation, ozone pollution, and salinity, which will all negatively impact our agri-food systems. I will not be covering those today. I will provide as many examples from rice wherever I have the data. So this would be the outline of my talk today. First, I'll present the current status of global food security, malnutrition, stunting, and crop yield losses and food waste. Then I'll talk about climate change and climate variability. What has happened in the past, what is happening now, and what is going to be the future. Then I'll go through the impacts of high temperature stress and sensitive stages. And I'll dig into the mechanisms causing reproductive failure and lower yields. I will then provide the thresholds and the temperature response curves for major grain crops. We will then discuss the interaction between temperature and carbon dioxide. And then finally, I'll introduce you to the need for systems approaches, and I'll provide you examples of three, starting with sustainable agricultural intensification, what it means and what are its components, and then briefly highlighting the need of other systems approaches, one on broader One Health, and also using the principles of circularity in our agri-food systems. And all of these systems approaches can provide us with holistic solutions to address food, nutrition, water, energy, and climate security. And if time permits, I'll also provide a current status of funding, which is going on in the global south, and also examples from India, particularly which are focused on sustainable agricultural intensification. And I'll end with some concluding remarks. So let's first talk about the current status and issues related to food security, malnutrition, stunting, yield gains, yield gaps, and food losses. At the current levels of population, Globally, around 811 million people are malnourished or of food insecure. They do not have enough food to eat. And most of these people are in Africa, South Asia, and Southeast Asia. And in many countries in sub-Saharan Africa, about 15 to 30% of the population are food insecure. And this is not considering the impact of COVID-19 and other stresses associated with climate change, which will increase these numbers anywhere in the range of additional 20 to 30 million people. In addition to food insecurity, we are also facing serious issues with nutrition, particularly the micronutrient deficiencies. For example, in this visual, the iron deficiency, which causes anemia, is widespread. More than 30% of the children in most of Africa and South Asia are suffering from iron deficiency or anemia. In addition to iron deficiency, other deficiencies include zinc deficiency and vitamin A deficiency, which are widespread and must be addressed. Due to those undernutrition, food insecurity, and nutrient deficiencies, we have serious problem of stunting in children. Globally, about 144 million children under the age of 5 are stunted, which is about 21.3%. And the three regions, particularly South Asia, Eastern and Southern Africa, and Western and Middle Africa are the most impacted regions, where more than 30% of the children under the age of 5 are stunted, and this is unacceptable. This visual shows you the grain yield increases over time from mid-1960s onwards of the 4 major food grain crops: wheat, rice, maize, and soybean. The smaller embedded visual shows the relative yield gains of each decade. Particularly for all of us in agricultural sciences, this is very important. Although we have made huge advances in yield gains in the major cereal crops over several decades, especially in the mid and later parts of the last century, the yield gains after that period and the yield increases and the relative yield gains have been very low, less than 1.5%, and almost stagnant for the last decade or so, although there has been slight increases or positive things which are happening. But to meet the demand of growing population, we need to double the rate of gains of these major cereals and also legumes and pulses and other crops. We need to do this under the threat of climate change, which is intensifying both abiotic and biotic stresses. So this is a global yield map of major cereal crops. The yield gap is the difference between the attainable yield and the actual yield. I'm talking about the attainable yield, not genetic potential. Attainable yield means that some farmers in that region were able to produce that yield considering their biophysical and socioeconomic conditions using the best management practices. So it is possible to get those yields. As you can see, there are large and huge yield gaps in Africa. We are currently only achieving about 30% of the attainable yield. There is a lot of pockets in India also where we have large yield gaps. So there are tremendous opportunities to minimize this yield gap by using best agronomic practices that includes crop, soil, water, and nutrient management practices, among others. And of course, genetics and the use of improved genotypes play a significant role in that. Another big area we have an opportunity is to decrease the food loss and food waste. These numbers are staggering, ranging from 20 to 30 to 45%. This is the amount of food lost or wasted. Globally, we lose or waste about 30% of what we produce. This accounts to about 1.3 billion tons, with a cost of $1 trillion. And both developing and developed countries contribute equally or to the similar extent. In the developing countries, food is lost during the production, harvest, or during the storage. Whereas in developing countries, it occurs after the produce leaves the farm gate during transport, processing, and on our dinner tables. If we are able to minimize or eliminate these losses, we've already achieved half of the goal of increasing the food production by 60% to meet the needs of global population of about 9.5 to 10 billion people. So 30% is right there of that 60%. And it saves us also a lot of other natural resources while we are doing so, and also minimizes the greenhouse and energy footprint if you're able to minimize these losses. So that takes into me the topic of climate change and climate variability. Climate change refers to the longer term changes in the climate over several decades or centuries, whereas climate variability refers to year-to-year variability or within season variability or occurrences of extreme events, which is much more impactful on our agri-food systems. So the major greenhouse gases have been increasing rapidly over the last several decades. At present, in 2021, the average, the carbon dioxide concentration was around 415 ppm, which is much higher compared to what it was in 1960s, less than 320 ppm. And similar increases and trends have also been observed in other greenhouse gases, such as methane, nitrous oxide, and sulfur hexafluorides. These are extremely potent greenhouse gases, and even at small quantities, they will have serious impacts on our current and future climates. And almost all the places on the Earth have seen increases in global surface temperatures in the last 50 years. Particularly parts of the Northern Hemisphere have seen about 1 to 2 degrees Celsius temperature increases. The global surface temperatures in year 2020 were about 1.2 degrees Celsius warmer compared to the beginning of the last century. And that increase has negatively impacted our agri-food systems around the world. And now, about the climate variability. The last 2 years, the 2021 and 2020, year before that, 2020, we saw extreme weather events throughout the world. I'll just show you some examples from US. The summer of 2021, June to August temperatures were the hottest in the US history, tied with the Dust Bowl years of early 1930s. Similarly, year 2021, the percentage of land area experiencing drought stress was extremely high. These extreme dry and hot conditions caused many wildfires, which devastated the natural resources in many of the western parts of the US. It dried up many reservoirs around the country as well. And if you look at the year before, the 2020, US had the largest number of named storms. The number was set a record at 30. And similarly, if you just focus on Kansas, we observed the hottest December in 2021 on the record. So clearly, climate has been changing and have been extremely variable, and we are seeing greater number of extreme events. And we saw similar trends of rising temperatures in India as well. In India, 2019, we saw extreme high temperatures in several parts of central India. And even this year, April 2020, I believe we recorded the highest or hottest April in India over the last 120 years. And this is impacting the grain yields of major crops. Particularly this year, we might see negative impacts of high temperature or heat stress on wheat yields, which will impact our ability to export. And India has also seen increases in episodes of drought stress, which had significant negative impacts on crop production. So the drought-prone area in India has increased by about 57% since 1997. And about 68% of the crop sown area experiences some degree of drought each year, which is impacting more than 50 million people on an annual basis. And this was the data from 2016. But such events continue to increase in the last few years as well. In addition, sometimes when we get rains, it comes in extremely high intensity and at odd timings, which again negatively impacts the yield. Sometimes they come at the time of harvest or soon after harvest, damaging the yields. And it has happened this year. Early untimely rain caused huge losses on the fields of just when the crop was about to be harvested or was harvested and stored in the field. How does the future look like? Most of the models project that the new climates will not only have the mean higher temperatures, but also occurrence of extreme hot weather events, especially extremely hot for a short period of time, maybe a few days or a week. But these extreme events are much more damaging to crop production, particularly if they occur at the sensitive stages of crop growth and development. So when you look at the future projections again, by end of this century, it will continue. If we continue as business as usual, most of the regions around the world will see more increases in the mean temperatures. Similarly, the projected precipitation is going to be lower in some parts of the world, and in some parts of the world, you may actually receive more precipitation. But because of the evapotranspiration losses caused by higher air temperatures, this will cause more drought and agricultural and meteorological drought, which will negatively impact our agri-food systems. So now let's look at impacts of heat stress or high temperature stress. And as I already said, it is possible, or it's more likely, that we'll get short episodes of heat stress from few days to a week or even for a few hours. And we're already seeing that, and it will continue to happen in the future. If these stresses occur during the sensitive stages of crop development, it will negatively impact the yields. And I'm going to explain to you how it is going to do that. Before I go into the details, just wanted everybody to bring up to date about growth and development. As we all know, growth is increase in the plant height, area, weight over the time. Whereas development is changes in the stage from early to mid to late vegetative and reproductive stages. I'm showing you these stages in rice as an example. And all of you know there are three general phases: the vegetative phase, the reproductive phase, and the ripening phase. And within each of those phases, you have, after emergence, you have early to mid to late vegetative stages, same for reproductive phase, and then milking dough and physiological maturity. And not all of these stages are equally sensitive to stress. Some are more sensitive compared to other stages. We need to know which are those stages so that we can manage or avoid the stress during those stages. And in the early part of my career, I've conducted a lot of research on multiple crops to determine what is the most sensitive stage among the grain crops. In general, reproductive stages or reproductive phase of crop development is relatively more sensitive to high temperatures than the vegetative stages. And reproductive phase or state have many stages. And then, so what we did in this research was expose the plants to short periods of high temperature stress, anywhere from 5 days to 1 day and few hours. And I'm showing you the example of sorghum for 5 days, peanut or groundnut for 1 day, and few hours, 6 hours on rice as an example. This visual x-axis is the timing relative to anthesis. 0 is anthesis, negative numbers are days before anthesis, and positive numbers are days after anthesis. And I'm showing you the data on the floret fertility or the seed set as a percentage of control. So 100 is a control plants and under optimal temperatures, and then rest is expressed as a percent of control. As you can see clearly, exposure to 5 days of heat stress decreased floret fertility in sorghum when the heat stress occurred at anthesis or 5 to 10 days before anthesis, where you see the maximum reduction in floret fertility. And for a 1 day of heat stress in peanut or groundnut, as an example, the maximum reduction occurred at the time of anthesis and about 4 days before anthesis. And that's the time before anthesis, it's 4 days or a week before, coincides with sporogenesis, the microsporogenesis and the megasporogenesis, where the viability of the pollen grains and the viability of ovules is being determined. And then at the time of anthesis coincides with pollination, pollen germination, pollen tube growth, and fertilization. And for rice, even 6 hours of stress at the time of flower opening was enough to cause decreases in floret fertility. And furthermore, genotypes varied in their response to heat stress, and some of those genotypes were susceptible with greater reductions in floret fertility compared to others. And I'll get to a little bit more details on that. Especially now, next few slides will be on the rice. There is a genetic variability in response to spikelet fertility when they were exposed to high temperature stress in rice. This is just one example where I use the temperature gradient field tunnels to quantify the impact on several rice cultivars. I'm showing you the data again as a percentage of control, 30 degrees versus 35 degrees Celsius daytime. So there were large differences in cultivars. Some were highly sensitive, some were intermediate in nature, and some were tolerant. And we need to exploit this genetic variability. And most of the genetic variability or the major differences among the genotypes were reflected in the number of filled and number of unfilled grains. The tolerant genotypes had more filled grains, whereas the susceptible genotypes had fewer filled grains. This was as a result of loss of spikelet fertility or increased sterility. The main reasons were associated with pollen viability and the stigma receptivity or the reception of the stigma to the pollen grains. And one of the reasons associated with the tolerance, as represented here with increased or decreased spikelet fertility, was also associated with pollen production and the pollen reception by the stigma, as I just mentioned. The tolerant genotype produced a lot more pollen, and there was greater reception of the pollen on the stigma. I've shown you in the table here with an example of N22, which was a tolerant variety and had a greater spikelet fertility versus L204, which had the lowest spikelet fertility and very susceptible. They had very fewer pollen production. In addition, there was also greater pollen viability and the pollen germination of these tolerant cultivars under the high temperature stress. And I believe the N22 is a variety which is originated in India. And Krishna Jagadish, a former scientist from International Rice Research Institute, and his team did a lot of work on high temperature stress impact on rice. Here are some slides from that program. And they clearly saw a negative linear relationship between accumulated hourly temperatures above 33 degrees Celsius, where the spikelet fertility, where increasing the accumulated hourly temperatures above 33 decreased the spikelet fertility. And high temperature stress decreased the number of germinating pollen, as shown by Rang et al. And in this study, they had both heat and water stress, and also a combination of both heat and water stress as well. And they had 5 different cultivars there. The negative impacts of heat stress were greater. Drought stress also had this negative impacts on germinating pollen, but the combination of both drought and heat were similar, meaning that the heat stress was the primary driver in those conditions. Then now I would like to talk a little bit about escape mechanism. This mechanism was particularly important and was initially found in rice. This is called as the early morning flowering. In one of my first earlier studies where I compared various species of rice, I'm just showing you the data from the 3 species, the sativas, the glaberimas, and the interspecific crosses. So I observed that the glaberima types and the interspecifics were flowering very early in the morning. Not only they start early, but also completed their flowering very early from 7 a.m. to before 8:30 a.m. Whereas the other types were flowering starting late from 9 a.m. and went up to almost past 12. So thus they were escaping the early morning ones were escaping the stress, because in a typical day, the temperature starts to increase from morning, reaches the maximum during midday from about 11 a.m. to 12 p.m. or even solar noon around 2 p.m. So if a genotype is flowering early and completes the fertilization before the hottest part of the day, it is escaping the heat stress. And once fertilized, because from pollination to fertilization, it takes about an hour or less. So once the embryo is formed, it is relatively much more tolerant to heat stress. So that's a mechanism which has been observed in rice. And then there have been near-isogenic lines developed with this EMF or the early morning flowering trait. And then they compared that near-isogenic line with several other rice cultivars by Sasaki et al. So as you can see, most of the current cultivars were flowering very late, about 4 to 5 hours after the sunrise. And then that's the time when the temperatures were very hot. So if you are able to hot and all the anthesis pollination and germination and pollen tube growth were occurring during that hotter part of the day. So this near isogenic line with this EMF trait was flowering early. So there is possibility to utilize this, this trait in a breeding program to escape the heat stress so that the plants are a little bit more, will have more pollen viability and more formation of seed set will be higher. So we have seen that there is reproductive failure, right? So I'll now show you the mechanisms of reproductive failure and then why do we see lower yields under high temperature stress. And I'll provide an overview. I'm not going to go into the details, but if anyone is interested, please let me know and I'll talk a little bit more details on that. But before I go into the mechanisms, I just wanted to show you how rice compares with the other grain crops. This is a summary of my several years of my early career. So I've developed response curves for majority of the grain crops. I'm only showing you 4 of the cereals and three legumes, but I have similar data on several other crops as well. So we all know that different crop species have different optimal and ceiling temperatures, and they also have different response functions. Here, the response curves are for major legumes. I chose to represent the results of harvest index as it captures the impacts of all the components and what has happened throughout the growing season. We can see crops vary for optimum and ceiling temperature. And the ceiling temperature is the time when is the temperature at which there's no grain produced, the harvest index is 0. Among cereals, wheat is the most sensitive, which is ceiling temperature of 31 degrees Celsius, followed by rice at 35, sorghum at 38, and pearl millet with a ceiling temperature of 41 degrees Celsius. The pearl millet is the most tolerant among cereals. Similarly, among summer legumes, the dry bean was the most sensitive at 32, followed by groundnut, which is at 39, and soybean at 40. And I have not shown you the data for mung bean and cowpeas, which are even much more tolerant with the ceiling temperatures are about 42 degrees Celsius, which is similar to that of pearl millet. So with climate change, there will be a need to change or adjust our crop species selection based on the environmental conditions. So at least we have options. So now let's dig a little deeper into the reproductive failure or the mechanisms. So I've showed you that reproductive failure occurs. And now what are the mechanisms? There are basically five different mechanisms through which the viability of the gametes is negatively impacted, resulting in floret sterility or less floret viability, and in turn results in lower seed numbers and therefore lower yields. And the first mechanism is that high temperature stress increases reactive oxygen species, and it decreases the antioxidant capacity, which leads to the membrane damage. And then the membranes of the pollen and the ovules are also damaged, leading to the sterility. And then that ultimately decreases the fertility and seed set and seed numbers, and therefore the lower yields. The second is that high temperature stress changes the lipids in the membranes, particularly the oxylipids, leading to membrane damage and then leading to the loss of viability of the gametes. The third one is the stress decreases the pollen sugars due to the downregulation of the hexose transporters that starves the pollen grains and leads to sterility. The fourth one is changes in the pollen and stigma anatomy, where the exene wall of the pollen is changed or is thinner, and which causes, and also the rapid degeneration of the tapetal cells happens, which is very important for providing the nutrition to the pollen grains, and that leads to the loss of function. And the fifth one is related to reproductive morphology. So what stress does that is it decreases the dehiscence of the pollen, and then the dehiscence of the anthers and the pollen, therefore, and it also causes the desiccation of the stigma. And sometimes we also see that stigma exertion, I mean the height of the stigma is above the anthers, so the pollination is difficult, especially for those which are self-pollinating. And therefore, it also causes the poor pollen reception, and then all of these leads to the sterility. So that's an overall mechanism. I'm not showing you the data, but I do have data for it, so individually for each of these mechanisms. So now let's look at what is happening throughout the season from a whole plant perspective, just moving away from the gametes or or the flowers or anthesis. And here is some of the impacts, how the high temperature stress impacts the grain yield and the harvest index. High temperature stress causes increases in the canopy temperature and also results in higher vapor pressure deficits. And these leads to decreased photosynthesis. And so it also increases the respiration. So therefore, the net carbon available for the plant growth and partitioning is lower. And it also causes early leaf senescence and also shorter growing season as well, which leads to decreased biomass production. And as I already mentioned, it causes reproductive failure, leading to lower grain numbers. And high temperature stress also decreases the grain filling duration because of the rapid accumulation of the growing degradase. And also that results in the smaller grain size, because the shorter grain filling duration is not able is not compensated by slight increase in the grain filling rate. So altogether, the fewer numbers and smaller size, they result in decreased grain yield and therefore harvest index. So those were the mechanisms. So now, looking at the interactions, because when we talk about climate change, there is two components to it. One is elevated carbon dioxide, and then the second one is an associated increase in the temperatures. So we need to understand these interactions. I've talked about temperature stress, but now let's look at it. How does temperature stress interact with the carbon dioxide concentrations? And I'm going to show you some data on rice. This was the work done in Hartwell Allen's lab by Baker and Allen, and I was also working in the same lab a little after a few years. So to start with, we all know carbon dioxide is increasing, and then carbon dioxide levels are actually can increase the photosynthesis, especially at the current levels, because Rubisco is not saturated at the current levels. So increase in carbon dioxide will increase the photosynthesis and can lead to greater biomass accumulation. But when you look at from the grain yield perspective, that is more important for the farmers, you only see the benefits of elevated carbon dioxide on the grain yield only at the cooler temperatures, not at the warmer or higher temperatures. Because so the benefits of the elevated carbon dioxide due to increased photosynthesis and biomass are more than negated by high temperature stress. So there's no benefits of elevated carbon dioxide under high temperature stress. And I've observed this in many other crops as well. And I'll show you that information in a bit. But when you look at the impacts of elevated carbon dioxide on yield components, like the panicle numbers or the individual grain weights or a filled grains or harvest index, there is no benefit of elevated carbon dioxide on those traits. So again, no benefits of elevated carbon dioxide across all the ranges of temperatures that were tested. So, but whereas increasing temperatures will actually decrease the yield under both ambient and elevated carbon dioxide. Again, I saw this similar trend on multiple crops here, and I'm showing you another C3 example, a common bean. For this crop, again, increasing temperatures decrease the total dry weights, the seed numbers, the seed size, and the seed yield, and there were interactions of carbon dioxide. For total dry weight, elevated carbon dioxide increased the total dry weight across all the temperatures, whereas for seed yields, the CO2 benefits were only observed at cooler temperatures, not at higher temperatures. And for seed numbers and seed size, there were no effect of elevated carbon dioxide at any temperatures, and similar things were also observed in rice. And I have data for other crops, but I'm not going to show you because of the time constraint. Not only that, but in some species, particularly on common bean and on rice, we actually saw that the seeding temperatures for seed set under elevated carbon dioxide were about 2 degrees Celsius lower, which is shown in this visuals. On common bean is my data, and the rice is from Matsui. And again, the reason being here that under elevated carbon dioxide, the stomata closes. And when the stomata closes, the tissue temperature increases. And when the tissue temperature increases, it has negative impacts, and especially on the pollen function here, the seeding temperature was about 2 degrees Celsius lower under elevated carbon dioxide. So that was about the impact of temperature, the short term, the sensitive stages, the mechanisms, longer duration stress, and the mechanisms there, and interaction between carbon dioxide and temperatures. Let me look at the time. We are at 32-minute mark. So in the next 10 or 15 minutes, what I'm going to talk about is the need for systems approach to address these complex problems of food security, nutrition security, climate security, energy security, and managing our food waste and food loss. So we really need some novel and holistic platforms because agri-food systems are very complex and they're all the way from production to consumption. And we really need to be taking a holistic approach to address our resilience of our agri-food systems. So I'm going to put forward 3 approaches. First one will be sustainable agriculture intensification. The second one would be a broader One Health concept. And the third one will be on the circularity or circular bioeconomies. And I'll go a little deeper into sustainable ag intensification, but not too much. But for the other 2, I'll just introduce you to the concept. I know you already know all of these things, but just wanted to talk about it for the benefit of the early careers scientists. So let me start with the concept of sustainable agricultural intensification. Again, we have multiple issues: food security, nutrition security, climate security, and also a lot of inefficiencies in our systems. And as Dr. Sundaram said in the beginning, we have to increase the food production. We have under the threat of climate change, and also we have to take care of our environment and make sure that we are not releasing a lot of greenhouse gas emissions, or we're not causing any pollutions and not degrading our natural resources. So to combat both of this, meaning that food security and also climate security and not causing any damage, so that's why we need sustainable ag intensification. So the goal of sustainable ag intensification, or SAI, is to provide access to safe, nutritious, and healthy food at all times to all people from existing farmland without damaging our natural resources. The key point here is existing farmland, because we really do not have more land to bring into agriculture unless we cut down our forests, and we don't want to do that. And we also do not want to damage our natural resource and ecosystem health. It is a holistic approach, and it has multiple components. They can be broadly divided into 3. The first is genetic intensification, the box on your right, which is in the red color, which is focused on increasing yields, building tolerance to biotic and abiotic stresses, improving nutrition, and we are using both traditional methods and also novel genomics and gene editing tools. The second box, which is in the green, is agroecological intensification. That is more focused on diversification, efficient cropping and farming systems, improved agronomy, the soil and the water management, integrated soil water management, increasing resource use efficiency, integrated nutrient and pest management strategies as well. And the third one is the socioeconomic intensification, which is focused on developing markets, creating new markets, policy interventions, understanding the barriers of adoption, building social capital, and creating enabling environments, and also institutional building. And the use of innovations in digital tools, the geospatial tools, the artificial intelligence, mechanization, precision agriculture, entrepreneurship, and private sector engagement is critical, and that cuts across all of these three aspects. So basically what it covers from a simplistic perspective is the interactions of genotype by environment, by management, and the social aspects, the G by E by M into S gems. So the whole approach of SAI is to provide a holistic platform. So that's a major aspect here. And the second is to consider a broader One Health platform. The Tri-Societies, the Crop Science Society of America, American Society of Agronomy, and the Soil Science Society of America recently developed their common grand challenge statements, and which reads as: drive soil, plant, water, environment system solution for healthy people on a healthy planet in a rapidly changing climate. So the use of broader One Health platform can bring us together. At present, One Health primarily focuses on humans and animals, and very little on the ecosystem, which covers primarily most of the other living organisms. But we all know that for better human and animal health, it all begins with healthy soils and healthy plants. And all healthy soils, healthy plants will lead to healthy animals and healthy humans. And together leads to healthy ecosystems, including our environment. And the circularity continues. And all of us are interconnected, both in positive terms and also in negative terms. In this visual, I've shown you some positive outcomes in green, and the negative outcomes are in the red. The positives includes nutrients, yield, nutrition, health, species diversity, ecosystem services, while the negative includes the pollution, the toxicity, the pests and diseases, concerns of food safety, degradation, natural resource degradation, the biodiversity loss. And COVID-19 also again reiterated the need for broadening the One Health concept and prepare ourselves for any such future disease outbreaks where it can cross one species to another species. COVID-19 spread from animals to humans. There are several others which can spread from between one species to another species, especially different biomes, including from soils and plants to animals. So we really need to look at the entire system so that we can avoid this or such future pandemics. And the third platform is related to circularity, with the focus on system redesign or system design and increasing the input and resource use efficiency throughout the system from production to consumption. We all know that there is large inefficiencies in our system, either due to loss of inputs or poor choices of inputs. We also have large nutrient losses, low nutrient use efficiency, issues with low water use efficiency, and more. And I also mentioned that we have a large food loss and food waste. About 30% of what we produce is wasted. So we have a lot of opportunities to improve the efficiencies and redesigning our systems. For example, for nutrients of water, if we use the principles of 5R, which is using the right source at the right rate, at the right place, at the right time, in a right manner. And that applies for both for water and nutrients. In addition, we can also eliminate the waste through reducing the use of it, reusing the waste, recycling the waste, repurposing the waste, and also better managing our residues. I do not have time to go into the details of each of these components, but this circularity can be applied for carbon cycle, the water cycle, the nitrogen cycle, and also can be methane cycle, especially all of these four are directly related to our agri-food systems. And I'm sure the rice production systems can also utilize some of these techniques in those production systems. So those were the three broader platforms. Now, in the next, we're at 40 mark. So in the next 5 or 7 minutes, so what I'll do is I'll briefly provide what our Sustainable Intensification Innovation Lab is doing. So we are funded by the Feed the Future initiative of USAID, US Agency for International Development. It is one of the largest innovation labs, and we have a very broad portfolio with systems research. We have active research, education, and outreach programs in multiple countries in Africa, in Asia, in Latin America, and Caribbean as well. I do not have time to talk in details. Please visit our website to learn more about our programs, but I'll just provide you an overview of that. Just a quick overview. We have several innovative multidisciplinary research projects in Asia, Africa, and Latin America, and Caribbean. And I'll just provide what are the topics which we are working, and then you can go and find more details. In West Africa, in Niger and Burkina Faso, Ghana, and Mali, we are focused on crop-livestock interactions, looking at dual-purpose crops focused on pearl millet, sorghum, and cowpea. In Asia, we are focused on rice fallows, use of cover crops, use of short-duration, high-nutrient-dense crops, and also commercial home gardens, and including the perennial wild gardens as well. And in East Africa, we are focused on cereal legume systems and water management using the principles of conservation agriculture. We're also using solar-powered drip irrigation systems and the locally available mulch. And then in addition to those research projects, we have 4 consortiums and 2 centers of excellence. And you can see the multidisciplinary nature of it. One of the consortium is on appropriate scale mechanization with the University of Illinois, and which is developing new and improving the designs and new mechanical tools all the way from planting to harvesting to threshing, irrigation, and also forage chopping and mixtures as well. That's where lots of engineering folks are involved in that. And the second consortium is Digital Tools and Geospatial and Farming Systems Research Consortium that is within our own university, which is focused on developing digital tools and appropriate land management land use systems, and use of remote sensing tools for quantifying plant health, and also using crop simulation models to evaluate the impact of various management strategies, and also look for our opportunities for climate adaptation and mitigation strategies to overcome biotic and abiotic stresses. And we also supported and created a policy research consortium to identify what type of policy interventions are needed, how do you track the impact, and what have worked in the past, and what are some of the metrics and indicators needed for those. And we're just wrapping up that consortium. It was with Rutgers University. And we also collaborated with International Fertilizer Development Center, IFDC, and created a soils consortium. And in addition, we have 2 centers of excellence, actually 3. One of them is a Center of Excellence in Sustainable Intensification and Nutrition. We call it CSAIN at the Royal University of Agriculture in Cambodia, which provides opportunities for research and education in Cambodia. And the second one is IREACH, which is Innovation and Research Education Advisory and Coordination Hub, which is in collaboration with CORAF, which covers the Western and Central African nations. And more recently, in 2021, we just got funding to establish a Center of Excellence for Mitigation, Adaptation, and Resilience to Climate Change in Haiti. I've not added that to this visual, but I'll update it soon. And we collaborate and fund large network of scholars. We support about 120 scientists, more than 100 organizations, which represents national agriculture research extension services, universities, private sectors, and international organizations in our host countries. It also includes 18 US universities, 7 CGIAR organizations, and the partnerships continues to grow. We thank all of our partners for doing excellent research, particularly developing innovations that would benefit smallholder farmers. I'll just, okay, I'm 45-minute mark, so maybe 3 or 4 more minutes. So I will mention briefly what we're doing in Asia. As I said, in Asia, we are focused on rice and rice fallows. In Bangladesh, we are working on testing and scaling high zinc rice in the polder and coastal zone in the south. And we observed that these short duration, high yielding, and nutrient dense, particularly high zinc rice, are yielding more than the traditional varieties. And because of the shorter duration, it also gives us an opportunity to add a legume or a vegetable crop soon after the harvest. And if you're mechanizing both the front end in terms of planting and transplanting, and also harvesting, we can those activities could be done faster. We have an opportunity to introduce a new crop. And then also introducing fish and rice integration is also a significant part of it. And these rice were developed by Bangladesh Rice Research Institute. I know even your institute has developed several of these biofortified varieties, which will be very useful. And in Cambodia, we're focused on rice fallows. We are using cover crops in rice fallows, sometimes as a relay crop, in collaboration with Penn State and CIRAD and University of Tennessee, and also the Royal University of Agriculture and our own Center of Excellence, and partnership from the University of Parangbang. And the 2 legumes which are as a cover crops which we are focusing are Silosanthus and Centrosema. And both of them are biological nitrogen fixation. And we are using mechanization tools for using roller crimper to incorporate these biomass. And then we can actually direct seed rice. And most of this is upland systems. We have seen multiple benefits from improved soil health, decreased nitrogen cost, and also minimizing the soil erosion and nutrient losses. And then for paddy rice, we are looking at options of green manure. And one of the classical and good example is the Sunham Pennsylvania as well. So, and then in addition, we're also looking at providing a small land for commercial home vegetable gardens where they are producing indigenous vegetables, which is very nutritious and also fetch higher market value. And then rice mulch is commonly utilized for these vegetable systems. Lastly, before I close, I would like to introduce you to the commission. This was an International Commission on Sustainable Ag Intensification, which was established in 2020. It has about 23 to 26 people on that, and I was honored to be part of this commission. The goal of this commission was to identify and recommend priority areas to strengthen the agricultural systems to meet some of the sustainable development goals and their climate needs. We are just wrapping up this commission, and most of the reports are available on the website. So I would encourage you to look at them. We have developed many policy briefs which are available freely. So the two questions which we were asking, I will only talk about the two items which I chaired, at least one of them, and then another one is still coming. One of the first question which we asked was, what is the status of investments in innovation in agriculture, particularly in the Global South, and who are funding this? Dalberg Asia produced this report. So they showed that about $60 billion are being invested annually on innovation in agriculture in the Global South. But of this, only 7% aligns explicitly with environmental outcome, and only half of that has social or human goals. So only 3.5%, which is extremely low and needs to be changed if we were to meet the sustainable development goals and keep the greenhouse gas goals of the Paris Agreement under 2 or under 1.5 degrees Celsius. Now you may ask, where is this funding coming from? So about 60% of those funds are coming from government, 25% is coming from private sector, 10% is coming from donors and development partners, and very little, less than 5% coming from private equity and venture capitalist companies. And the same is true for India as well. So the Indian, in the case of India, so again, the same 4 buckets: Indian government, the bilateral, the multilateral, international philanthropies and venture capitalists, and private entrepreneurship. On the government side, about 10% of the total funds are invested in innovations. And only 4.5% of that are aligning with sustainable ag intensification. From bilateral, about 60 to 70% funds are on innovation and only 6% on SAI. From philanthropy, 90% on innovations and only 3% on SAI. Whereas the venture capitalists mostly focused on innovation, but only 1% of that is focused on SAI. So this really needs to shift if we were to make a case for mutual goals of climate security and food security. And the second question was, what is the innovation gap to remove the hunger and meet some of the climate goals? And this report was developed by Mark Rosgrant and his team at IFPRI, International Policy Research Institute. They found that the total gap, very conservative figures, the total gap was about $15 billion per year. If you can invest or reiterate this amount yearly up to 2030, we can minimize the hunger below 5% in most of the regions in the world, except for sub-Saharan Africa, where we can reduce it to 11%. Not only that, we can significantly decrease the greenhouse gas emissions, agriculture use by 10%, deforestation by 925,000 hectares. So clearly a modest investment of $15 billion focused on research and scaling and remove hunger and address some of the climate goals. And for more details, please see the report. So let me conclude now and summarize my thoughts. Hopefully, the last 50 minutes, I was able to convince you that climate change and climate variability will have negative impacts on grain yields of our major food grain crops in most of the regions. Even short episodes of 1 day or even few hours of abiotic stresses, if they occur at critical stages of crop development, will cause negative impacts on yields. And food grain crops are sensitive to critical stages at key stages of crop development, particularly at gametogenesis, flowering, and early grain filling stages. The yield decrease due to stress are primarily due to decreased grain numbers and the grain size, and to a little extent, lower biomass production, shorter green leaf area duration, and lower partitioning to grains. More importantly, the beneficial effects of elevated carbon dioxide are more than negated by high temperature stress. There are no benefits of elevated carbon dioxide at high temperatures. There is genetic variability in the current breeding programs, but it is very limited. Thus, we need to explore systematically larger germplasm collections, including wide species for tolerance. And then, of course, we need to develop high-throughput phenotyping tools and molecular and biochemical markers to screen larger germplasm collections to identify these traits, and then match it with the genotyping so that the breeding can be done rapidly. And interactions with multiple climate change factors are not well understood. I only talked about temperature and carbon dioxide, but we have issues with water, radiation, vapor pressure deficit, photoperiod, and on top of it, also the biotic factors, the pests, the diseases, the weeds, the interactions. These interactions are not well understood and needs attention. And we need a better understanding of the impact of climate change factors on grain nutritional qualities. And then we need to combine the yield potential with stress tolerance with the grain composition perspectives. Then transdisciplinary research and approaches like sustainable ag intensification, broader one health and circularity must be used. And the collaboration between crop scientists, agronomists, soil scientists is critical and needed. We need a convergence of both biophysical and social sciences to understand the barriers of adoption and create enabling environment. And lastly, current investments in innovation, which have environment, human, and social dimensions, is extremely low, and that needs to be reversed to meet the goals of sustainable development and climate action. With that, I would like to thank everyone. And if you would like to learn more about our activities, please visit our website and follow us on our social media. Again, thank you so much for giving me this opportunity today to speak to all of you. Thanks. I'll be happy to take any questions if you have.
Thank you, sir. Thank you for enlightening us on various phases of this topic on crop response to key climate change factors and how to mitigate them for food and nutritional security. I think you have given a bird's eye view of this very good, very large topic, sir, and with in-depth analysis on investments and giving a roadmap for future research and investments. Thank you very much, sir. I think there are very many participants who would like to interact. Now I invite Dr. Tuti to moderate this session. And sir, will you be able to take up some questions?
Yeah, I'm available as long as you are available.
So Dr. Tuti will moderate the session, sir, and please raise your hands so that sir can interact in this session. Thank you.
Thank you, madam. Indeed, it was a great presentation. It was very much excellent. And there are few questions and queries. Let us go one by one. Dr. Arun Shankar from Krida, actually, he raised a few points. Yeah.
Good morning, Dr. Vara Prasad. As always, it's been. Are you people able to hear me?
Yes, we are.
And as always, it's been fantastic listening to you. And every time you talk, I learn something new. In fact, the first part of your presentation was very much in the area of what I work here. The past 2 years, I've been working on temperature tunnels and pearl millet in elevated carbon dioxide and 3 different gradients of temperatures. And I'm focusing specifically on chlorophyll fluorescence and electron transport pathway. The question I wanted to ask you was my results, the first year of experimentation. I've been seeing that there is an increase in the efficiency of that is FV by FM quantum efficiency and various other electron transport pathways which are favorable towards photosynthesis is increased in elevated carbon dioxide in a C4 plant, that is pearl millet, and the temperature also is not that high. It is about close to 40 degrees. So I just don't know actually why this is happening in a C4 plant where carbon dioxide concentration mechanism is already there, and it is not going to gain much in terms of elevated carbon dioxide in terms of dry matter production. But still, I see this increase in the photosynthetic efficiency in the PS1, that is a light reaction. Can you throw some light on this?
Sure, absolutely. Thank you very much again. Very good to see you. And I also see that Dr. Sai Prasad is here. Thank you very much for joining. Great question again. We saw similar results on sorghum, which is also a C4 crop. And then my inquisitiveness is that the crans, especially the crans anatomy, is not fully developed in younger leaves. So if you are measuring at a leaf, depends upon where you are measuring that for the quantum efficiency. If it is a younger leaf and the crans anatomy is not fully developed, that might be the reason why you are seeing that increased photosynthesis even at elevated carbon dioxide in those conditions. But once the leaf is matured and when you're at a later stages, you may not see that effect.
Oh, that's interesting, and thank you again. Just another one. Actually, another thing is that I'm also seeing that I have developed a method to quantify oxygen evolution, that is OEC damage. And I'm also seeing a certain degree of OEC damage, but that is made up by elevated CO2, in the sense that I see that elevated CO2 to an extent is compensating for the loss created by a little bit of heat, not the higher heat, the first grade.
That's right, and especially for pearl millet, because it is a very relatively heat tolerant crop. So even at lower temperatures, the elevated CO2 will compensate, not only at a cellular level, at a leaf level, and at a whole plant level as well.
Thank you very much. It's always nice listening to you.
Thank you.
Thank you very much, sir. The next queries come from Dr. S.P. Sai Prasad, sir. Actually, he wants to know about the climate resilience, the capacity of various old varieties, mega varieties, as well as the new released varieties. So, sir is there, he can put forth his question.
Congratulations, Dr. Vara Prasad. And we really enjoyed your talk. Just I want to know that we are telling that the climate change is increasing and temperatures are increasing, but still we are not able to old varieties, replace the old varieties, whether they are having more adaptable to these climate changes, or the new varieties are more adaptable to the climate change in practical terms.
Yes. Well, let me take that question and talk about resilience. So I think, in my personal opinion, people may differ from me, but I believe that some of the old varieties are much more resilient to climate change, but they do not have the required yield potential or the genetic opportunity, and they may not be even responding to the inputs which we are giving. So in terms of, those, like for example, N22, it's an extremely old variety, right? So, but it may, so that is the most heat tolerant, or escaping the heat. So that's just one example, and I've seen the same thing happening in wheat as well. So the old varieties, they're more resilient, but they're not adaptable to the input management practices which we are giving, and therefore, they don't have the genetic potentials. So we need to utilize those old varieties in the current breeding programs to transfer those appropriate genes so that we continue to increase the rate of gain of yields.
Thank you very much, Dr. Sir.
Thank you very much, sir. We could able to see Dr. Padmini Swami, NRRI director. She is there. She has also given good comments about the presentation. If madam is there, she can ask any queries or questions.
Thank you, Dr. Vara Prasad. Really, you had a very excellent and brilliant talk. It's really mesmerizing. And those who are working, particularly nowadays on climate change, particularly high temperature and CO2 enhancement. So it is really a boost to them to use in their future scientific career. So I have nothing to ask, only Dr. Sai Prasad, what he asked, I was also having in my mind, the old varieties, still they are prevailing. Still farmers are continuing with that. Maybe they are having the better resilience. Of course, yield level, of course, it is not reducing, but in the current varieties, whatever we are developing nowadays, they are having high yield potential than the older ones. But the older ones, maybe the they are having the resilience to sustain in the current changing climatic scenario. So I got some of my questions answered by you, what Dr. Sai Prasad was asking. Yeah, and it's excellent. I again congratulate you also.
Thank you.
And some other, my young scientist, they may have some query. I think from Anjani Kumari, he was asking about ozone, I think, elevated ozone. One of my colleagues from our institute, you can see if you can throw some light. Thank you very much.
That's great. Thank you so much for your comments. And again, I did not go in deep into the mechanisms and other associated items. And you're right, this is more for an early career scholars so that they get excited and they need to think about systems research and how crop scientists can work with agronomists and soil scientists, ag economists, social sciences. So that's the main message which I wanted to give is think in terms of transdisciplinary research and break down the silos. And specifically trying to answer the question of Dr. Anjani Kumar, he was asking that how does crop responses under elevated ozone scenarios? Yes, ozone is increasing, especially at the lower levels where crops are impacted. And then again, the impact of ozone is going to be because of the spots which it develops, the photosynthetic capacity of that leaf is going to be decreasing. So therefore, you see rapid senescence and lower green leaf area duration that will negatively impacts the yield. And not only that, ozone will also cause tomato closure, which will also decrease the photosynthetic capacity. That's at the lower level. But if you look at a stratospheric level where you need the ozone, and then where, because of the decreased ozone layer, the ultraviolet UVB radiation is going to come in, and then that will also have a negative impact. So ozone at the lower levels, having ozone at the lower levels is a bad thing, but at a higher level is a good thing because it's protecting the coming of the ultraviolet radiation. Again, there's quite a bit of work done on ozone, and I don't know if ozone pollution will decrease the gamete fertility or not, but probably the result of lower yield is because of its impact on photosynthesis because of the rapid senescence and the decreased green leaf area duration.
Thank you very much, sir. Dr. Anjani. Dr. Anjani, you can put your question.
Sir, thank you, sir, for throwing light on the crop responses of under elevated ozone scenario. Sir, basically, what would be the effect of elevated ozone on greenhouse gas emission from rice soil? Please tell something about this also, sir.
I have not studied that component, so I wouldn't want to take a guess on such a topic, which I have really not done work on impact of ozone specifically from the perspective of greenhouse gas emissions from the rice fields. But I would assume that because of the productivity decreases, there might be a negative impact on yield. And of course, when you have the negative, when you have such impact, the efficiency of water use, efficiency of nutrient use will be decreased. So it indirectly will have less input or resource use efficiency, which will have negative impacts on both from a carbon, water, and nutrient footprint perspective.
Okay. Thank you, sir. Thank you, sir.
Thank you very much, sir. Yeah, Dr. Parveen Kumar, Director ICAR-IIRR is among with us. So we would like to hear from him also, his thoughts about this talk and this about climate resilient technologies.
Good to see you, sir. Good to see you.
Yeah, thank you, Dr. Mangaldeep. At the outset, sir, I would like to compliment and extend my hearty thanks to Professor P.V. Vara Prasad ji. Sir, I listened to you in November 2021 in International Agronomy Congress, and at that time your talk was really excellent. So today when I got the message that ICAR-IIRR is conducting your webinar, I could not stop myself. I was just connected at 9:45 15 minutes before the scheduled time. So today also, as earlier your talk was really mesmerizing, highly informative. I learned many new things, how exactly the high temperature is impacting the physiology of the specifically rice crop. How many new things I able to understand the complexity of the high temperature along with the carbon dioxide. I was also not aware. I was thinking that when CO2 will increase, definitely that photosynthesis will increase, but that photosynthesis if increase at the higher temperature is not so beneficial. Or sir, apart from the plants, actually since I am working for the coastal agriculture, the higher temperature is also increasing the sea surface temperature. That is also decreasing the marine fish catch, because those fishes, because of higher temperature, their oxygen concentration is getting down. Because of that, the marine animals is going down, and in general, the catch of the fishes is decreasing. So higher temperature is not only bad thing for the crops, it is also damaging the marine life as well. Sir, your talk was really excellent. I am really happy to hear you, and thanks to Dr. Mangaldeep to just say my comments. Again, thank you very much.
Thank you, sir. Thank you, sir. Nice to see you too. And you're absolutely right. I think what I showed you was just one crop, but we haven't talked about the impact of this climate change on aquatic and fisheries, both controlled and uncontrolled ones. So there's huge negative impacts there.
And sir, I also invite you, whenever come to India, please visit Goa. It will be our pleasure to host you, sir. Here.
Thank you. And one item which I really didn't cover was the impact of these temperature on different diseases and pests, because the disease life cycle, the disease triangle of the pathogen environment intensity is going to be negatively impacted. You're going to see the host plant susceptibility increasing, and also the virulence of the pathogen is going to increase. And then not only that, in terms of entomology, the locusts and fall armyworm, those are all because of the climate change. What has happened is the Indian Ocean dipole temperatures have changed, and that was in 2018 and 2019, which resulted in the 2020 and 2021 locusts.
Yes.
And of course, didn't talk about the weeds as well, because most of these weeds are going to be much more tolerant to these, and their interaction with the crop plants is going to be very negative. And not only that, the herbicide efficacy, efficacy is also going to be negative. And there's one question by Suvarna Rani: if the grain size decreases, the identity of a particular variety will be out. Please share your thoughts. So for the yield, there's three components which makes the yield: plant numbers, individual grain size, and the grain numbers. You multiply these through these three things, you get yield. So as I showed you clearly that the seed numbers or grain numbers are significantly decreased by increasing temperatures, particularly if they happen at the time of flowering or a few days before flowering. And then the grain size decreases because it impacts the especially at the rapid grain filling stages, the smaller grain sizes because of the decreased grain filling duration. So identity of the variety is not really based on the grain size, but I think some people say that higher temperatures will increase the rate of grain filling. But all of my research, what it has shown is that the slight increase in the grain filling rate does not compensate for the loss caused by grain filling duration. Then, and I also mentioned about the impact of this high temperature on the grain nutritional qualities, which I didn't have a chance to talk to you today, but especially the antioxidant content, the micronutrients, and there's carbohydrates versus protein. There is a trade-off between them as well. So that's another area which is important and needs attention.
Yeah, thank you very much, sir. Another couple of questions are there, sir.
So I see the next question is from Dr. Padma Kumari Garu, and she's saying that in the situation of changing climates, should we concentrate on ambient temperature or microclimate data, especially with reference to temperature and humidity? That's a great question again. So I think we have two components of climate change, right? In addition to, if you take out carbon dioxide, we're talking about two things here, temperature stress and the drought stress. And most of these happen simultaneously. So especially for rice, let's talk about heat stress. Under heat stress conditions, if you have soil moisture available, or if you have the irrigation opportunity, so there's a whole system called canopy temperature depression or cooling. So the plants have the ability to absorb the water and cool the canopies and avoid the temperature stress. So that's one possibility. And then when you're irrigating, the microclimate around the plants will be, the cooling effect will be there, so you may not see the negative impacts of temperature stress. But if you do not have an option of irrigation or microclimate cooling, then you would see the combination of both drought and heat stress. Under those, the best strategy is that, because under high temperature stress, the vapor pressure deficit is going to be high. So if you're wanting for heat and drought tolerance together, or at least some combination of heat stress and some relatively moderate water stress, you should be screening varieties for vapor pressure under high vapor pressure deficit. It's going to give you the combination, both. And then, but always, how do you provide that microclimate coolness is going to be important. If you're irrigating, then again, make sure that if you're irrigating in the early morning when the flowers are anthesis, high humidity, can also have a negative impacts in pollination, especially on crops like cotton, and I believe it's also true for sunflower and other crops. I'm not sure what happens with rice, but I think so we need to be considering those as well. If you have water, climate cooling is an option, but it's not always the case because it comes at the expense of water.
Yeah, Dr. Rajendranagar sir from NRRI Cuttack.
Thank you, Dr. Vara Prasad. It's a very nice talk. I enjoyed it. So I am having one queries to you. So climate's resilient agriculture, climate smart agriculture, that rests on three pillars: productivity, adaptation, and mitigation. A country like India, when we prioritize climate smart agriculture, we give weightage to productivity, again, adaptation, and mitigation. So would you kindly suggest if we prioritize for our farmers, how much weightage in this context need to be given mitigations and adaptation and productivity? Listen, adaptation and productivity.
Okay, thank you so much for mentioning that. You're right. We are focusing on productivity, adaptation, and mitigation of climate change. On a short term, we need to be focusing on adaptation. Adaptation to climate change is very important because mitigation is going to take a longer time frame. Because to minimize the greenhouse gas emissions and overcome the current other issues, it will take us time. But adaptation can be done through multiple ways: the genetic adaptation, the management in terms of adjusting your planting dates, choosing the right duration of the variety, and matching the G by E by M interaction. So that needs to be the case. But I would like to take a step back and then actually talk a little bit more about, we need to go beyond productivity, right? So in my opinion, there are 5 domains. When farmers are making decisions, they're looking at 5 things: productivity, economics, environment, social dimension, and human conditions. So these are the 5 domains. There's always a trade-off happening between these. You may be very productive, but not economically viable. Maybe negative impact on the environment. May not be nutritious. And you may not have the resources or social capacity. So for any of our innovations, whether it's genetic innovations or agronomic or any innovations, we need to be looking at trade-offs of these 5 domains: productivity, economics, environment, social, human conditions. Because when farmers is looking, they're not just looking at one component of it. And that's why we have issues of adoption and scaling, because we are only looking at one component. Productivity is extremely important, don't get me wrong. It's required, it's needed, but we really need to be thinking a little bit more beyond that, on top of productivity.
So the corollary to this question is, when we are going to prioritize this technology in the context of a developing world and a developed world, the weightage need to differ, because the burden of mitigation cannot be given to the solely the farmers of developing countries or least developed countries. If we are planning the same, prioritize the same kind of technology for developed countries, they can compromise the productivity and give more weightage to mitigation and adaptation. And the reverse is the case in case of developing a least developed country. Would it be appropriate in the context of your equity in climate justice?
Yes, absolutely. I think you're right, because let's look at, that's why I would say that total factor productivity. That's what we need to be looking at. And then per capita is what needs to be accounted for. So especially for, you're right that for a smallholder farmer or countries who are still developing and there's still a large amount of yield gaps, and where the input, especially in organic fertilizer use, is very low, there is still opportunities to increase that. When I showed you that a graph of yield gaps, right? Majority of the sub-Saharan African countries is a huge yield gaps. And if you look at just the amount of fertilizer they use in their crops, it is less than 25 kilograms per hectare. Go back to 1960s. 1960s, India was using about 10 kilograms per hectare. Africa was also using about 6 kilograms per hectare. And now India has gone up to almost 100 or just past 100 kilograms per hectare. And Africa is still moved from 6 to 25 or 30 kilograms per hectare. So with that type of inputs, on average input on the crops, how can you expect that you get more yield? And there is not enough organic material in their farms to add additional nutrients because of the competition for the crop residues, for example, for livestock feeding, for fencing, for roofing, and sometimes wildfires and termites get it. So there's a lot of competition related to that. So what I'm saying is that, therefore, we need to focus both on productivity and environment. That's the concept of sustainable ag intensification. I'm not just saying sustainable agriculture or intensification. The combination has to be there for the developing countries, because we are not there yet. We still have 60 to 70% yield gap, and that would require more inputs, but of course also would require more efficiency of the inputs. We don't want to make the same mistake. I mean, India did a fantastically great from 60s to 90s. We did not correct the course in late 90s or early 2000 to make sure that we are focusing on efficiency rather than maximizing the yield. That's why we are seeing some negative impacts on our water quality, issues with water, and other associated items. We don't want to repeat the same thing in Africa, but so we have to be a little bit more cautious. And my focus, we should talk about regenerative agriculture. Not only, I mean, in future is going to be not damaging, actually making some positive change is required. And it can be done. It can be done. We all work together.
Thank you.
Thank you, sir. And another question is there from Dr. Suvarna Rani. Grain size decreases, the identity of a particular variety will be doubtful. So she needs some clarification in this.
I think I responded to that. I already responded to that question, unless she has additional things. I did talk about that. Okay. And there was the thing which, there's another question from Sujata.
Yeah, another
What's
one
the role of minimum temperature?
Yeah. What is the role of minimum temperature, which is raising above 25 degrees constant?
That's correct. So if you look at, when you talk about when you talk about warming or increasing temperatures, there's two components to it: daytime temperature, the nighttime temperature. And there's been significant evidence that is the nighttime temperature which are increasing at a relatively larger rate compared to the other daytime temperatures. So most of those effects which we are seeing in my studies which I've shown, I've increased both daytime and nighttime temperature simultaneously. But we have tremendous amount of data, particularly for rice and wheat, which shows that the nighttime temperatures are worse. And why they are worse? Because if you look at nighttime temperature, the respiration rates, which is happening both at day and nighttime, whereas photosynthesis is only happening at the daytime. And then the pollination, the pollen germination and pollen tube growth is only happening early morning. Or actually, in case of pearl millet, anthesis is happening in the nighttime as well. And so is the case with, same is the case with cowpea as well. So in my opinion, the nighttime temperature has much more damaging effect than compared to that of the daytime temperature.
I think this much queries are there, sir. Everything has been answered. So now it is time for Dr. R.M. Kumar, Dr. Padma Kumari Madam to further progress. Yes.
Are there any more questions? Please raise your hands. If there are no questions, we, as a token of our love and appreciation, we have a small letter of commendation for a beloved speaker. I request Dr. R.M. Kumar to do the honors.
Thank you.
So this is a certificate which would be sending to Dr. Vara Prasad Garu in the virtual mode. I think all nice things should come to an end. So, sir, R.M. Kumar sir, should we go for the formal vote of thanks?
Just 1 minute, madam. Just okay.
Yeah.
Before vote of thanks, sir. Actually, our director has to conclude your webinar, but he has to attend some very important event in PJTSA. He has conveyed his sincere apologies. I'm not going to conclude any of your. This is one of the best webinars which we had, and it has been given very good insights for all the rice workers who are being. I'm seeing that who are been attending. They were throughout the session, so they were very much following this webinar, and they are very attractive, and then they could able to gain a lot of knowledge, because since we are working on this sustainability in our most of our programs in Indian Institute of Rice Research also, we need your guidance in future also, sir. We expect that your presence during June, July also, we would like to have a physical interaction with you during that time, so that we can learn many more things from coming visit. So thank you very much, sir. It was an excellent, and I think I expect that everybody to give a big hand for our renowned speaker, Dr. Vara Prasad Garu. Thank you, sir. Thank you very much. Padma, I think I request Dr. Arun to go for formal vote of thanks.
Yes.
Dr. Arun.
Yes, sir. Thank you, sir. Thank you, sir. Thank you, Mahendra, sir. Is it audible, ma'am?
Yes. Go ahead.
You're audible.
Yeah.
Go ahead.
Yeah, good morning. Good morning, everyone. It is indeed a great pleasure to propose formal vote of thanks for this webinar on very important topic of relevance. Today, we had one of our distinguished speaker, Dr. P.V. Vara Prasad, Director, Sustainable Intensification Innovation Lab, University Distinguished Professor, Kansas State University, who has given a excellent talk on crop responses to key climate change factors and need for systems research. Having an eminent speaker and a scientist for their task helps in bringing clarity on an important topic that deals many complex issues. With his vast research experience, not only he connected all the dots, and finally, flamboyantly, he portrayed a solution in the form of a systems approach. Thank you, sir. On behalf of the ICAR-IIRR Seminar Committee and all the participants of this webinar, we are very happy to thank you for accepting our request and giving this excellent talk. I also take this opportunity to thank our beloved director, Dr. R.M. Sundaram, for always giving his support and encouragement for organizing such important webinars continuously on a regular basis. And thank you, Dr. Padmini Swain, ma'am, Director NRRI, for being there always in our webinars. So that symbolizes your encouragement and also your support in all our initiatives. Thank you, ma'am. And also we thank Dr. Parveen Kumar, Director, ICAR-CCARI Goa, for actively participating in this webinar. Thank you very much, sir. And also I thank all the IRR scientists, NRRI scientists, and our ACRIP cooperators and other scientists from CRIDA like Dr. Arun Shankar sir for active, AK Naik sir, Dr. AK Naik sir for actively participating in this webinar, both in Zoom and also in YouTube live streaming. Special thanks to Dr. Aram Kumar sir from SAR Society, and also from Dr. APPK, AP Padmakumari ma'am for actively organizing such webinars. Thank you, ma'am. AKMU for providing this logistic support, and thank you each one of you for actively participating. Thank you very much.
Thank you.
Thank you, sir.
Thank
We
you,
are going to close this session. Thank you. Wait, Padma, go ahead.
Thank you. Thank you, one and all.
Thank you.
Thanks.